Hello everybody, this lesson is going to be about intermolecular forces. These are the forces that hold molecules next to each other. So we're going to learn how different molecules start to interact with each other based on whether or not they're polar or nonpolar. So a couple of terms. Intramolecular forces are the forces that hold the atoms together inside the molecule. So those are things like ionic or covalent. Those are pretty strong. We typically call those bonds, right? An ionic bond, a covalent bond. An intermolecular force is what holds two or more molecules near each other, or potentially what causes them to repel from each other. Okay? These are much weaker. We don't call these bonds, we call them forces. Okay? You can say that this is an ionic force or an ionic bond, a covalent force or a covalent bond, but we're really going to call these intermolecular forces, and that's going to remind us that they are weaker than a full bond. There are lots of different types. We're going to look at the main ones. We're going to rank them from weakest to strongest, and then we'll learn about each type. So the weakest intermolecular force that we're going to have is called London dispersion force. Then comes something called dipole-dipole. It is a little silly that we say it twice, but that is the real name. Remember, a dipole is what you get when you have a polar molecule. So this is trying to remind you that you have two things that have poles on them, right? The strongest is called a hydrogen bond, which is a terrible name because it's not really strong enough to be considered a bond. It should have been called maybe hydrogen force. That might have been a better one, but unfortunately this is the name that stuck. You've probably heard this in biology class before, and I just want to make sure that everybody understands that even though we say hydrogen bond is strong, it is still very weak compared to a real bond, okay? So it's a bad name. We should have called it hydrogen force or something like that. Sometimes you may see the phrase van der Waals forces. Van der Waals forces are the sum of all London dispersion forces and dipole-dipoles added together. The reason they get lumped together into one big category is because they're very hard to measure individually. And so because it's hard to measure what portion is coming from London dispersion and what portion is coming from dipole-dipole, what they can do to make things a little bit easier, they just measure the cumulative force from both of these combined. So if you hear the phrase van der Waals forces, you should just know that they're combining those together. Let's look at London dispersion forces. Sometimes we abbreviate that to be LDF because nobody wants to write that whole thing out. It's really, really important that you understand that these are very, very weak and they are temporary. They are not permanent, okay? So what ends up happening is you have something that's non-polar and the electrons are spread out nice and evenly all over. But remember that the electrons are not locked in place. Remember that they move around randomly. And there is a possibility that in any one instant, just by sheer chance, the electrons might accidentally end up clumped together on one side of the molecule or atom more than the other. And so what ends up happening is for a very brief, instantaneous moment in time, you get a tiny little dipole happening. And so it can have an attraction with something else that has a dipole. So the thing is, the electrons are not going to stay there, are they? They're going to keep moving. And so this very temporary, very weak, unequal distribution of electrons will go away, right? So for a brief moment in time, there's a tiny little dipole, but then they go back to being spread out, and then that's gone. And so it's very weak because it's temporary, because it's not a huge discrepancy between where the electrons are. It's just that they're a little bit uneven for a moment in time. So these are not very strong. We see these in everything. Everything has London forces, okay? Anything with electrons is going to have a moment where the electrons are not balanced out perfectly over the whole structure, right? But in a non-polar molecule, this is the only thing we have holding molecules near each other. So if we have something like I2, non-polar, for a brief moment in time, they will be able to be attracted to each other, but not for very long. So really important that you understand everything has London forces because everything has electrons. The bigger your molecule, the more London forces you would have. This gets a little bit tricky to measure because it isn't really the mass per se, it's more the surface area, and it also has to do with how tightly bound the electrons are, and we say if it's, polarizable or not. So this is a pretty tricky one, but almost always it will work out so that the bigger the molecule, the more London forces you have. So the more electrons you have, the more chances you have of them being unequally distributed at a given moment. So kind of the quick and dirty way to guess, what has more London forces would be to count up your electrons. Just realize that's not perfect, but that's kind of the best we can do at this level. So for example, C8H18 and C3H8 are both nonpolar, but C8H18 will have more London forces because it's a much bigger molecule, there's way more electrons, there's more chances for the electrons to be unequally distributed. So there's more chances for it to get a little bit of a pull once in a while. Moving on past London forces, we get dipole-dipole. So this is going to happen in polar molecules, and these are permanent, okay? Because the molecule itself is polar, that pole will never go away. So these forces don't go away like a London force does. So for example, you can see here HI, this will always be polar. And so there will always be some kind of an attraction between them. Remember, we have our partial negative and our partial positive, and they are electrostatically attracted. It's not as much as a full bond, but it's more than nothing, right? And so it's something that we have to think about and take into account. Are these molecules going to be held towards each other? They're not bonded to each other, but they are being attracted to each other. Next is hydrogen bonding. Hydrogen bonding is actually a type of dipole-dipole. All it is a very strong dipole-dipole. But it's so strong and it's so important to biological functions that they give it a special name and they treat it as if it's its own category, even though it is just a dipole-dipole. So we say that a hydrogen bond has to have H-NOF lone pair. Okay, kind of a funny little phrase. I'll say it again, H-NOF lone pair. What that means is you have to have a hydrogen that's bound to either a nitrogen, an oxygen, or a fluorine. Okay, have to be attached to one of those three. It can only be one of those three because these three are very electronegative. We have to get a big difference in electronegativities here. And so nitrogen, oxygen, fluorine are going to be the only things strong enough to get a strong enough dipole, strong enough pull on those electrons. The other piece is this lone pair here. The nitrogen, oxygen, or fluorine has to have a lone pair attached to it. Okay? So it's really important that you look at the Lewis structure, not just the formula. You may have hydrogen and nitrogen, but if they're not connected to each other, it's not a hydrogen bond. If there isn't a lone pair on it, it isn't a hydrogen bond. People get these problems wrong a lot because they only look at the formula and they don't look at the actual structure. So maybe make a little note to yourself to always draw a little Lewis structure for a problem asking if it's a hydrogen bond or not. So if you look here, we have two water molecules. We have the hydrogens bound to the oxygen. Oxygen has some lone pairs on it. We end up with one end down here, the hydrogen side, that's a little bit partial positive, and this side is a little bit partial negative. And so what ends up happening is the partial negative side ends up lining up and being attracted to the partial positive side of another water molecule. We draw it with a dashed line because it's not a full bond, it's not that strong, but it's strong enough to really impact the properties, so it's something we want to take into consideration. So this would be an example of a hydrogen bond because I have a dipole-dipole, but more specifically, I have hydrogen bound to an oxygen. The oxygen has lone pair on it, so this ends up pretty negative, and this side is pretty positive. So it makes a much stronger force here. So we end up with the partially negative part of the lone pair on one of our NOF, we say NOF, N-O-F, NOF atoms, and it's being attracted to the hydrogen end of one of those bonds. We have a pretty partial negative over here, that partial positive over here. We end up with an electrostatic attraction. For those of you that like flowcharts, you can see this represented in a flowchart. Is the molecule polar? Well, if it's not, the only thing you can have would be London forces. There's nothing else to have those molecules interact with each other. It's just London dispersion forces, LDF. If it is polar, then you have to look to see, hey, do we have any HNOF going on here? Anything with hydrogen connected to nitrogen, oxygen, or fluorine? And really, really big, really, really bright color, something super obvious, we should have a little note here that says with the lone pair on it. It's not enough to just have the bond. You have to have it with a lone pair as well. So be very careful about that. If you don't meet these criteria, then you just have a normal dipole-dipole. You also have London forces, okay? Because remember, anything with electrons has London forces. The dominant force is the dipole-dipole. We don't often ask you to list all of the forces present. We usually are just asking you to list the dominant force. If you did meet the criteria over here, that means you have a really strong dipole, specifically called a hydrogen bond. Remember, it's a type of dipole, it's just extra strong. You also have those London forces, right? So now let's look at what can happen to the properties because of these forces. If molecules are going to be drawn towards each other, that's going to affect their properties. So for example, you probably learned in biology that water has a very high surface tension. Sometimes they do a little activity where you have to count how many drops of water you can fit on a penny so that you can see that the surface tension is pretty high. The reason for that is these molecules are holding on to each other with these weak forces. They're weaker than a bond, but they're still strong enough to change the properties. Water also boils at a fairly high temperature. 100 degrees is pretty high, which is good because we would not want all the water on Earth or in our bodies just boiling away at room temperature, right? That would be pretty bad. And so the fact that water has such a high boiling point means that we're able to really have life on this planet, right? So that's why biology is so, interested in these hydrogen bonds, is because even down to the fact that we're water-based, right, is really impacted by these forces that we have. So here's some properties that you can look at. Boiling point, melting point. Viscosity is how thick something is. If it's very viscous, it's very thick, like syrup is viscous. Surface tension. When you have more IMFs present, all your forces are higher, so you're going to get higher properties, higher boiling points, higher melting points. It's going to get more viscous, higher surface tension. Miscibility is whether or not something mixes. We have a phrase, we say like dissolves like. Polar substances like to dissolve and mix with other polar substances. Non-polar things like to mix and dissolve with other non-polar things. If you've ever seen, the classic water and oil, they don't mix. Water's polar, oil is non-polar. You can figure out if something's polar or not by mixing it with something else that you do know the polarity of. If they mix, they have the same polarity. If they don't mix, they have opposite polarities. One's polar, one's non-polar. I'm going to show you just two more special examples of IMFs that are related to biology that they want us to just make sure you don't forget about. And one of them is DNA. The reason why our DNA is able to unzip, if you remember, it's in that helix shape, and it has to unzip so that it can be replicated, right? It has to be, copied over. Well, the reason it can do that is because these base pairs are held together with hydrogen bonds, okay? They're not ionically bonded. They're not covalently bonded. Those are too strong. They're hydrogen bonded. It's enough to keep them together when you want, but they're weak enough for us to be able to take them apart and unzip these strands more easily. So I like to say it's the difference between gluing something together and using Velcro. Okay, the hydrogen bond is Velcro. It will stick when we want it to, but we can easily peel those strands apart from each other. These are some examples of the base pairs that are in the DNA. You probably remember being told that A pairs with T and G pairs with C and they line up, and that's the, combos that can match. But sometimes they don't show you why that's true. And the reason why is because these pairings allow the hydrogen bonds to line up correctly. So in an AT pair, you can see here, we end up with 2 hydrogen bonds that line up nicely. In a GC pair, we have 3 hydrogen bonds that line up nicely. Now, I would never ask you to draw these base pairs or know what the base pairs are. I mean, that's bio, that's not chem. I don't know these. I'd have to look them up. This is not what I do, right? I do chemistry. But if somebody showed me these pictures, I know the chemistry well enough that I could look at them and I could highlight and show people where these hydrogen bonds are happening, right? You look for your HNOFs, right? and that's going to help you find those hydrogen bonds. Proteins are another example of this. You probably learned that there's different folding patterns to proteins. We can have the alpha helix, kind of like our DNA, but you can also get beta sheets. Remember, there's like primary structure, secondary structure, tertiary, quaternary, where these complex molecules start bending and folding around, and hydrogen bonds start causing them to kind of crumple up into these shapes, and that's how we get complex things like our hemoglobin. These complex shapes, like this, end up having very specific places that can perform very specific roles in our body. So for example, in the hemoglobin, we have these sites that have iron on them that can help our blood cells carry oxygen to, our cells around our body. They can bring carbon dioxide back to our lungs. If this is not folded properly, if these hydrogen bonds get messed up for some reason, we no longer can perform the functions that we need in our body to be healthy. Okay, just a couple of really specifics they want us to remind you of from bio class. All right, the last part of this is going to be bulk solids. Bulk solids is what we call things where we have a huge, overwhelming combination of intramolecular forces and intermolecular forces on a big bulk scale. So usually you have some inter and some intra, but in a bulk solid you have so many of these things happening in all three dimensions that things end up very, very strong, stronger than you think they should be. So these bulk solids have very high melting points, very high boiling points, right? Because they're being held on to so strongly. Metallic substances are bulk solids. Ionic lattices are bulk solids. And something called network covalent is a bulk solid. If we were going to be very generic and rank these from weak to strong, metallic would be weaker, then ionic, then network covalent. But all of these, even the weak metallic ones, are still typically stronger than anything else you would see, any other forces, because we have so many happening. So an ionic lattice is what you get when you have an ionic compound that's kind of structured like a crystal in all three dimensions. Metallic does the same thing in terms of, kind of lining up into a structure like this. And then remember that the electrons kind of detach and flow freely like a sea of electrons through all these atoms. And that makes it pretty strong. Network covalent is when we have covalently bonded atoms that are kind of connected in such a complex network that it ends up stronger than you would think. Carbon is one thing that does this. Silicon also does this. One of these is diamond and one of these is graphite. You probably have, an intuition that diamond is stronger than your pencil lead, right? This one over here on the left is diamond. You can see how many bonds we have going on. We have all sorts of intra and inter forces happening here. Very, very, very strong substance. What you may not realize, though, is that graphite is also very strong. It is also a network covalent. Inside these sheets, right? There's a lot of bonding happening. This is very strong in here. The thing that makes graphite have some of the properties we are used to is the fact that these sheets are stacked upon each other. There are intermolecular forces holding these sheets together. That's what makes graphite kind of slippery, right? If you think about it, when you're writing with your pencil, right, some of the graphite is getting sloughed off onto your paper. I don't know if you've ever done that thing where you kind of scribble your pencil on your paper a whole bunch and then you rub your finger in it to get it all over your finger, and then you try to make little fingerprints with the graphite. I feel like in middle school, everybody always does that, right? Well, the reason it feels so slippery on your hand is because graphite actually has a lot of lubrication properties, because these sheets can slide past each other. That's why it feels so slippery. They actually use it in machinery to keep the gears from sticking, right? It lubricates the gears so that the machine parts can, move past each other nicely. And because it's so strong in the sheets don't break down. They slide past each other, but you're not going to, deteriorate the actual sheets, which is why it's such a good substance to use. So the last slide for this PowerPoint is just showing you an overall ranking from the weakest IMFs to the strongest IMFs. Now, you have to understand this is a horrible oversimplification, right? There are definitely times where you have an ionic substance that might be weaker than something that's metallic. You might have a situation where there's so many London forces that it's stronger than something with a hydrogen bond, right? But we have to simplify it enough that we can learn and practice and progress through our chemistry class, right? If we got so hung up on every exception, we'd never get through anything. So if I ask you to rank things based on IMFs, you're going to take it at face value and you're just going to rank them like this. You're going to look at the structure, you're going to decide what type of IMF it has, and then you use that to rank them. If you start looking up things like boiling points online, you may get all confused. You may say, but this has a higher boiling point, but it has a weaker IMF. But you got to realize that, this gets very complicated very fast. And so we are going to be ranking things in a pretty simplistic way. Okay. So just follow your little chart here and you'll end up with an answer that shows that you understand the concepts that we're working with. All right. Okay. That's it, everybody. I hope that was helpful. Bye.